Hello, everybody. Welcome to the INC seminar. Gautam, who's my friend from the Redwood Center in Berkeley, and also working in Ehud Isaac's lab in Berkeley as well, and who I've interacted with in Lisbon. He's a very fun guy to hang out with, and also a very smart guy. He's currently a professor at the Claremont colleges, and I'm really looking forward to hearing his interpretations of how to think about neural waves and computing with them. So without further ado, very excited to hear from you, Gautam.
Thank you, Mike. And in the spirit of what I understand your chalk talks to be, in lieu of chalk, please ask any questions that come up for you during the course of the talk. One phenomenon I've been really interested in is brain waves and what they tell us about the brain. And this is what this talk is about. So when you measure electrical activity from the brain, depending on the nature of your electrode, you can pick up very different kinds of signals. So if you put an electrode on the scalp, you pick up what's called the EEG, which essentially shows you these wave-like patterns of activity. And if you go, that tends to be on the order of millions of neurons. And then if you stick the electrode a little deeper, you get into the brain tissue itself, and you can pick up what's called the local field potential, or LFP. And that also has this kind of wave-like character. And now the resolution seems to be on the order of tens of thousands of neurons in terms of how many neurons sum coherently to generate what you pick up on an LFP recording. And then if you go deeper still, and you're very close to the cell body of a neuron or inside a neuron, then you see intracellular activity. And specifically, you can pick up what are called spikes, these binary action potentials. And most of the field is very interested in characterizing brain activity and brain function using these spikes. They seem to provide the highest resolution of information. You're picking up single neurons, and these neurons seem to be tuned to features of the world. And so you can get a very kind of high bandwidth image of the contents of this brain region. But the local field potential and EEG also carry their own sort of information. Namely, if you look at these brain waves, you see that their shape, their frequencies, and their amplitudes tell you something about the state of the organism. So for example, if the organism is very excited, then you tend to get faster and smaller waves. And as you relax more and more, the waves tend to be larger and of lower amplitudes. So this is all to say that the shape of the brainwave can give you some indication of the behavioral state of the animal. And what I was really interested in understanding or studying is whether these waves could give even more granular information about the contents of the nervous system, a la spiking, the way that people use spiking and the activities of single neurons to infer brain contents. Can we use brain waves to do something similar? And so this is the high-level question that we're interested in. And I'll go over what we're doing with this in 3 different chapters. And really, if I get through the first 2 chapters, I'll feel happy. So if we have time for the 3rd, we'll go into that, but feel free to ask any questions as they come up. So the region and signal that we're studying is called the theta rhythm of the hippocampus. It's a very well-studied brain rhythm, and it's many of the studies are done in rodents. So here you have a rat, and this yellow region is its hippocampus. And when you stick an electrode and look at the LFP in the rat hippocampus, you see that when the animal starts running, as indicated by this increase of speed, you see that the LFP starts generating these relatively periodic triangular waves. And these triangular waves happen at around 8 hertz and are called the theta rhythm. And that's different than what you see when the animal is not moving. And we'll get into this more later on in the talk as well.
Gautam, is your LFP filtered at all, or is it a raw signal?
So most of the analysis we'll be doing is at this 8 hertz band. And so what we do is we take a recording that's at 1250 hertz as collected by our collaborators. And then we downsample that to about 40 hertz, which is ample for capturing the properties of the rhythm that we're interested in, which is theta. We also apply a high pass filter at 2 hertz, and that gets rid of some of the 1 over F signal, which tends to dominate the LFP. So roughly we're going between 2 and 10 hertz, and that picks up mainly this theta rhythm. So one thing that is really interesting about this rhythm is that people have started conceptualizing the function of this rhythm within the hippocampal circuit. So namely, if you look at the theta rhythm and you simultaneously look at the spiking of a neuron in the hippocampus, the neurons of the rat hippocampus are often referred to as place cells because they activate when the animal occupies specific locations of its environment, then you see the cell tends to fire at a specific phase of the rhythm, and that phase advances as the animal runs through the so-called place field, the place in which this cell activates. So as the animal is running through the place field of the cell fires periodically at an earlier and earlier and earlier phase, a phenomenon known as phase precession. And one consequence of phase precession is that if you have many cells that have place fields that follow each other in the environment that the animal is navigating, then within a single theta cycle, you have a rapid run-through of the activities of these different cells based on the phase they tend to fire, which in turn is based on their relative position along the track. So essentially what you have is, over the time course of seconds, a playing through of these 4 neurons, and at the time scale of 1/10 of a second, a much time-compressed version of the same code within single cycles. So you can kind of think of the theta rhythm as chunking little snippets of trajectories representing the animal's path. Any questions about this so far?
Yes, is in dark room, or is all the sensors are open? The eyes, the hearer.
Yeah, the recordings we're looking at, it really depends on the experiment. What we're doing is in the dark. Rats are very tactile, so there might be like olfactory and other information that they're using, but the basic idea is that a lot of these codes are determined by the animal's estimation of their own location as they move through the environment, and as corrected by specific landmarks such as the reward ports. So you can say that visual information can refine the location of these place fields, but it's not necessary.
So I have a quick question. So if the mouse stops at the let's say the center of location D,
Yeah.
will the place cells associated with that specific spot continue to fire even if it has stopped? And if they do continue to fire, then for how long?
Yeah, they continue to fire. They're less high fidelity than when the animal is running. And kind of apropos, relevant to our talk, the theta rhythm becomes much sloppier in this time. So this tight coordination seems to kind of break down when the animal is stopped. As far as I know, there isn't any time limit on how long these cells will remain active. Like the animal can be there for many dozens of seconds. I haven't found recordings where they're sitting there for much longer, but I believe they exist. They keep activating for longer periods of time as well.
Okay, and my second question is, if you were to check back on this mouse in like 3 weeks or a 1 month or like 2 months, do these place cells stay the same? Are they still associated with the same sites or locations?
Yeah, so these are rats, and rats and mice have similar properties, but they're a little different. So with rats, the place fields can be somewhat preserved over, let's say, the time span of a day or two. But over longer periods, the population code seems to drift. Now, I think the working knowledge in the field is that while the individual place fields drift, the relationships between place fields seem to be more persistent across time and across environments.
Thank you.
Yeah.
Is there main theta rhythm at all correlated with the speed of running?
Yes, so it becomes higher amplitude and faster, slightly faster as the animal starts running faster. Okay, so here we have this model of the theta rhythm serving as some sort of clock. One issue is that the rhythm is actually much more complex. If you dip an electrode and look at the rhythm at many different locations within the hippocampus, you see that actually the shape of the wave, its timing, its amplitude, its waveform changes depending on where you are looking at the signal. And here is a movie showing the same thing from a two-dimensional array of electrodes that's been inserted in the hippocampus. And this is showing you the theta rhythm while the animal is running through space. And what I want you to kind of focus on is the fact that the amplitude of the signal changes depending on where you're looking at it, as well as the timing of the signal, the way that the wave is flowing through the tissue. And when we first saw this, we got really excited because whenever there's this kind of variation of form, there's the potential for the form to carry some information that the circuit cares about. And that's what we were interested in doing. Can we read out information about the rat's behavior from the shapes that are embedded in this traveling wave?
Good question.
Yeah.
How local are the connections in this tissue on the left?
So the tissue, they're both showing a cross-section of the hippocampus. Now, the place cells are fairly local within the tissue. Let's say with this layer that you see over here is a layer of cell bodies, and a place cell might span kind of this region on average. Now, they're connected by interneurons, many of which are much broader in their reach. They kind of might actually traverse the entire hippocampus. So as far as what's generating the wave, it seems to be mostly synaptic inputs and quite distributed synaptic inputs. If not by virtue of the field of the synapse, by virtue of the fact that these cells are very connected by one or two connections to the entire hippocampus.
Thank you.
Yeah. Okay, so in order to read these waves out, we took inspiration from radio communication. The idea there being that a radio signal is one where you can embed a relatively slowly changing signal in a much faster changing oscillation. So when you multiply these two, you get a waveform that can be transmitted and picked up by a radio. And for example, if you have the so-called amplitude modulation, you can look at the envelope of the signal to reconstruct the original waveform. And now the brain is much sloppier than a radio transmitter. So how do we go about applying this approach to a brain rhythm? So first we really benefit from using the framework of complex numbers. So imagine this is a brainwave that we're recording. And what you can do is use a so-called Hilbert transform to essentially infer a time-shifted version of the signal. And when you look at this new twofold dimensionality of a signal, a two-dimensional signal instead of a one-dimensional signal, it traces out an orbit in a 2D plane. And this is great for us because now we can define the so-called phase of the signal, the angle between this point and the x-axis, as well as the amplitude of the signal, how far it is from the origin. And so in order to then apply this kind of decoding of a brainwave, imagine you have a wave that you're recording from an electrode. And this is just a schematic. Let's assume that this wave increases in its amplitude, and then at some other point, it shifts in its phase relative to some sort of reference. And the reference that I'm showing you here is this black line. That could be seen as the carrier wave. So what we can do is we can convert both of these signals to a complex number, a two-dimensional signal that now can be visualized as a corkscrew in time. And now what you can see is that this corkscrew has a period where its amplitude is really high and a period where its phase seems to be lagging that of the carrier. And what we can do then is subtract the phase of our signal of interest from the phase of the carrier. And what you're left with is this slowly changing signal. So this is an operation that's called demodulation. And what you see then is now we've removed the fast changing component of the carrier wave, and you're just left with the signal that is preserving these modulations of amplitude and phase, which we believe carry the relevant information. Any questions?
I have a question. So I understand the extracting the amplitude and the phase and why that's useful. I don't understand what you gain by going from the 1D to the 3D. Like, it seems like you could get the amplitude and phase changing information from that first curve at the top.
Yes, so in fact, this signal is created by this signal. So it's just a way of making phase more explicit. You're right, the phase is here as well. The problem is that if you just look at this signal itself, you can have two positions of equal value that have very different phases. So this is just a more convenient way of extracting phase because you're disambiguating these two values that have the same coordinates.
Okay, thanks.
Yeah. Okay. So we're still missing one thing, which is we don't know what the carrier wave is when we are dealing with brain signals. So the way we dealt with that is applied what's called PCA, principal component analysis. And essentially what this does is it looks at the signal that's measured across all the electrodes, and it says, what is the kind of most dominant component across my measurement channels? And essentially you can think of that as being an average of all of the electrodes that produces a single component that is the most prominent component of your signal. So this is what we're using as a proxy for our carrier wave. And here what I'm doing is I'm just showing you the real components of the signal from 8 electrodes. We're recording from many more, but for visualization purposes, I'm just showing you 8. And I'm just showing you the real component. Then what we can do is subtract the phase of these complex valued signals from the phase of our carriers. And when you do that, you're left with these demodulated signals that preserve any changes in amplitude at individual electrodes, as well as any changes in phase. So we're demodulating each electrode's signal using this signal that's common to all the electrodes. And that allows us to see patterns of amplitude and phase across these electrodes. And now that we've removed this fast changing oscillation from the signal, we can use kind of basic supervised learning, linear regression, to predict where the animal is within its environment. Namely, we train a different linear model that represents each position along the track. So we know that position is what the hippocampus cares about. And so that is our kind of candidate for what these patterns in the brain waves represent. And so each one of these decoders represents a particular location in the environment. And we can train these linear models and use the most active feature as an estimate of the animal's current location. So in our task, the animal's running back and forth on a linear track. It's getting water rewards. And our goal then is to predict where along the track the animal is. And we can do that by using linear regression on this demodulated signal.
Excuse me, but that means that every point in the trajectory have to be different kind of stimulus to the rat, right? In order to detect different points, because it's the same pattern for all the trajectory. The stimulus is the same, and then it's not possible to detect the difference.
That's right. That's right. So we know that in the rat hippocampus, different cells activate at different locations. So you can imagine, let's say there's 100,000 neurons in place cells. Some fraction of them will activate at different locations, and different populations will activate at different locations. So the basic idea is that different populations of these place cells give rise to different phase amplitude patterns in the LFP. And that's what we're able to use to localize the animal's position.
I mean that it's not natural behavior. It's only you made the stimulation, different kinds of stimulations through the path, right?
So this is a real recording. So this is a recording of real data as an animal is in a fairly artificial environment. You could imagine that a much more realistic environment is foraging in the wild or something. But a lot of the studies are done in this very constrained environment, but this is real data that we're using to decode.
So, how repetitive is this hallway? Like, how easy is it for you to tell, or how easy do you make it for the rat to tell how different one spot is in the hallway from another spot, and how long is the hallway? Well, you say it right here, but yeah, like how distinguishable do you think the hallway is, and what happens if you make this hallway have some sort of repeating pattern? So it becomes very, very hard. When you make it very long, it becomes very, very hard for the rat to tell where along the path it is. We start to see repetitions in this pattern.
Yeah, so it depends on the region you're recording from. So if you record from what are called, known as the entorhinal cortex, then you see cells that fire at a repeated interval in the animal's environment. They're known as grid cells. So the longest tracks so people have done studies, for example, in bats that happen across many, like tens of miles. And in rodents, there's studies that use tracks that are maybe 10 times longer than this. And in the hippocampus, you don't seem to see any repetition. And you can imagine these animals that are really expert navigators have very sophisticated ways of knowing where they are in even very large spaces, a lot of which rely on what's called dead reckoning, keeping track of your own behavior to estimate where you are in your environment.
Thank you.
Yeah. Okay, so now in order to kind of evaluate the quality of this code at the level of the brain waves, we can compare these decoders to what are in the field more traditional spike-based decoders. So you can look at the pattern of the spiking activity in place cells, and you can use that if this is the rat's trajectory in black as the animal's running along the track, you can use the cells patterns of activity, the population spiking, to infer or estimate the animal's current position, and that estimate is shown in red dots. And so if these red dots overlap significantly with the black line, it means you have a good decoder. And when we compare the decoding of these waves, we find that actually the quality of our estimate seems to be as good, if not better than, the estimates we get from simultaneously recorded neurons from the multi-electrode array. And so this, for us, kind of was very exciting because it suggested that these very slow and coarse-grained waves, which people typically dismiss as being relatively non-selective, are actually very informative when it comes, or can be very informative when it comes to the contents of a neural circuit. Okay, so what I've been showing you is mainly from the periods of time when the animal is running. And so these periods are kind of unique because the theta rhythm is very highly rhythmic. And so you could imagine it might be a little easier to pull out information from these circuits because all the neurons are synchronized in some way. But we also know that if you look at the hippocampus in other periods, for example, when the rat is still, you see much more irregularity in the pattern of the oscillation. And this is not only true in the rat. If you look at other species, for example, bats or humans, you tend to see signals that are more like this, as well as if you look at non-hippocampal regions, such as the cortex. So we were really interested in knowing how well the technique would work when we look at these kinds of signals that are somewhat more less regular. And already, if you look at our decoder as the animal is running, you see that the quality of the decoding seems to be a little worse when the animal is still. So this is a period when the animal is still, the theta rhythm is more irregular, and our green dots, which are our estimates of the animal's location, don't seem to overlap so reliably with the animal's actual position. So this suggests that there's some kind of limit to our ability for decoding. And so this is what I've been working on more recently, is how can we recover information from these brainwaves? Can we? And so for that, we've been collaborating with Albert Lee's lab, which until recently was at Genelia Farm. And what was really exciting about their data was that they were using many more electrodes, up to 256 electrodes implanted in the hippocampus. We thought that maybe with more electrodes, we would have better access to the underlying signal. Also, they designed a task where the animal spends very extensive periods of time collecting reward. So this allows us to have lots of data from these time periods where the animal is still and the LFP is less rhythmic to really validate our decoders. So now if we look at the neural activity while the animal is running, what you're seeing is the activity at something like 32 different channels out of up to 256 channels. On the top, you see the first principal component, which is the average signal. And on each subsequent line, I'm color coding the signal at one electrode based on its phase relative to the carrier wave, what we were calling the carrier waves. And so you can see that these signals seem to be fairly consistently rhythmic and fairly coherent across the electrodes. Now, when the animal is still, this rhythmicity, as we saw previously, becomes much weaker, and there seems to be somewhat less coherence across the electrodes. And so now what we can do is see how well we can decode the animal's position. Namely, what I'll be trying to do is decode which of these 3 arms is the animal currently on. So that's kind of a classification task that I'm using. And comparing the quality of my decoder when the animal is running in these areas marked in blue, as well as the periods where the animal is still in these orange boxes, where the LFP becomes much less rhythmic. So with the animals running, what I'm showing you here is the confidence of the decoder that the animal is on one of 3 arms. And this tall black line means that the decoder is very confidently indicating the animal to be in the correct arm. So where the animal is where the decoder thinks it is, and it's very confident. And there's a few instances where it seems to break down, but in general, it's very confident in the animal's location. But when the animal is still, when the rat is still, the confidence really deteriorates. It becomes much less confident, and there are longer periods of time where the decoder actually thinks that the animal is on a different arm of the maze. And so we wondered how we could, what was causing this drop in decoding? Any questions?
Yes, in the case that is running, what happened in the neighborhoods? What is the environment in that case? Because they changed something, right?
Would you say it again?
You can see the minimums when the rat is running.
Yeah.
No continuous, they have a minimum over there. And then what happened? What is the environment in this point?
What happens when the decoder fails?
Oh, so you're talking about these particular moments here.
Yeah. What
Is that,
changed?
is that when he's switching arms, for example?
No, so this is actually, it's a good clarifying question. So this is actually during one run on one track. So this is the animals on this track, and we're trying to predict which track the animal's on. And so when you see the decoder fail, it's actually in the middle of the track. And I don't know why this is going on. My prediction would be that this is a window of time when the place cells from a different track happen to be active for whatever reason. So we know, for example, that the animal can have remote replay of parts of the track that it's not inhabiting. The prediction would be this is happening during these times, but I don't know.
If you downsample the same signal, do you think that would average out, or?
Yeah, so if you downsample, then you're essentially averaging the pattern that's present along more of the track, and that would remove these periods of error.
It'd be really interesting to know if it is remotely playing which parts of which tracks it's remotely playing during those little clips of time.
Yeah.
The track or something while it's in the middle, actually, something like that.
Exactly, and I think looking at what the spikes of the place cells are doing would be a good way of validating that as well. Okay, so then we can ask what circuit behavior might lead to the decrease of our decoding ability. So you can imagine that this is kind of a schematic of what the circuit looks like. So here you have different population of place cells. Different colors represent cells that are active in different locations. And it is thought that these cells can form assemblies where they can influence each other's activities corresponding to cells that are all active at the same place. And different colors can represent different assemblies that are active in different locations. It's also known that the hippocampus is driven by these presynaptic, these upstream nuclei. This one's called the medial septum, which projects to the hippocampus and seems to be kind of a pacemaker that drives rhythmic activity in this circuit. So when the animal is running and the rhythm is very strong, these assemblies can very easily entrain to the activity of these pacemakers. So here you see the rhythm, the collective activity of the pacemaker in black. In red, you see the activity of one assembly, and you see they're highly coherent. And so if we were to do some sort of demodulation, the carrier would be very reliable. We could decode where the animal is based on the patterns generated by this assembly. Now, one possibility is when the animal stops running, these cells, these presynaptic cells, become much less rhythmic, and these cells, the coherence among them kind of deteriorates. There's no pacemaker that's kind of binding their activity and generating a coherent oscillation. And so then you would imagine if these cells are incoherent, they wouldn't be able to generate an LFP that sums the coherent activity of all of these cells. And so in this case, we might imagine that we're kind of out of luck. We wouldn't be able to decode local field potential patterns with this lack of coherence. Another possibility is that when this presynaptic pacemaker becomes inactive and produces a very weak or irregular rhythm, these cells are able to maintain some sort of local coherence among themselves. And in that case, you would see this oscillatory activity, but it would no longer be aligned with what we believe the carrier to be. So for example, you might have periods where the phases of the two signals are aligned, but also periods where the phases are not aligned with each other. And so you could imagine if you're estimating the carrier based on the activity of these cells, it wouldn't be a very good reference frame for demodulating and decoding the signal. And so we were interested in kind of arbitrating between these possibilities, specifically if we could make a decoder that didn't rely on a carrier signal, it would support this hypothesis over this. Any questions? Okay.
The coding is not failing, is not probably is because the other cells is working at the same time, right?
I think the owl is having trouble picking up your question. Mike, could you repeat it?
I didn't understand it. Can you?
Yeah, because you know that you say the hypothesis is the coding fail.
Yeah.
The coding is not fail. That when the rat is static, is more in stress because it's looking what is the next. They don't know what is the next. So in the case that the rat is running, the rat know what is the next. It's running. It is they suppress many signals in the brain in order to run. And that is the reason.
Yes. So the reason we're calling this a failure, well, first off, you'll see more later on in the talk why it's a failure, but also the place cells do activate in a selective way while the animal is still and these rhythms are weak. So you can still decode the animal's position using these place cells, but you can't decode it using our LFP-based decoder. So the information is still there in the circuit. Okay. So earlier, I showed you an approach where we could use linear regression after removing the oscillatory component. Now, what we're going to try is train non-linear decoders, which are able to learn directly from these signals phase amplitude patterns. And so then each one of these decoders now is trying to detect a specific oscillation that activates at a specific location and represents a certain pattern of activity across the electrodes. And once we isolate these oscillatory signals without reference to this carrier wave, then we can look at which of these oscillations is the strongest to estimate the animal's position. And if this technique could work, we've sidestepped our reliance on our potentially noisy estimate of a carrier wave. So to do that, I've made a pretty simple neural network. This is a shallow neural network. And what it just has one hidden layer. The inputs of this network are the complex valued signal that's detected at each one of the electrodes. So here I'm showing you 6 electrodes. Our recordings might have on the order of 200. And then each node learns a set of complex valued weights. And when the phase amplitude pattern in the inputs matches the phase amplitude pattern of these complex weights, then this particular hidden node will be active. Now, in order to estimate the animal's position, we, just like before, we're trying to classify which arm of the maze the animal is on, but we have a hidden layer that has many more features that can self-organize a representation of the features that are relevant for predicting the animal's location at a coarse-grained level. So each one of these nodes learns a different phase amplitude pattern across the electrodes. And then what we do is once it's learned this, we strip away, once it's activated by this, this oscillation, we strip away the phase of the oscillation, just leaving the amplitude. And this tells us which phase amplitude pattern is on, regardless of the phase of this oscillation. So this step over here can be thought of as roughly corresponding to the max pooling operation of a convolutional neural network. So we want to detect a certain pattern, but we don't care about what phase that pattern is in. And then we can use these activations, which lack phase, to predict which arm of the maze the animal is on. Any questions about this neural network?
Why do you want to toss space?
Yeah, so we are keeping phase initially because we believe that the phase amplitude patterns are important, they're very informative. But once we know that a particular phase amplitude pattern is present, we don't care about the phase that the oscillation, that pattern is in. We just care about detecting that that pattern is on. So it's kind of like why people use max pooling in a convolutional net. Once you detect the presence of, let's say, a 0 on your image, you don't care about the exact location of the 0. You just care that the 0 is there. So that's kind of the equivalent of what we're doing over here.
Sure.
Okay. So when we have 3 output nodes, one corresponding to each one of these arms, and 3 hidden nodes, each one of the hidden nodes learns to activate in one of the 3 arms of the maze. And this kind of makes sense. The network is learning to self-organize a code so that each output is selectively activated by one of the hidden node features. Now, what's more interesting is what happens as you start increasing the size of the hidden layer. Then you start seeing that different hidden nodes activate at a specific location within the animal's environment, as if each one of these nodes is learning a place-selective cell assembly. So another way we can show this is being showing the activity of these hidden nodes as a function of space. Now I'm going to show the activity of these nodes as a function of time. So here are 3 different runs where the animal is running on arm 1, arm 2, or arm 3. Now, if you look at the theta rhythm at any electrode or by using PCA, you just see a rhythm that's always on. But if you look at the theta rhythms patterns that are learned by these hidden nodes, each node activates at a very specific moment while the animal is running. And if you arrange them by the order in which they activate, you see that they progress in their activations sequentially as the animal is running through space. And what's more is each one of these theta rhythms shows this phenomenon of phase precession as rendered in the color of this waveform. So the same kinds of behaviors we see at the level of place cells, we're seeing at the level of these phase amplitude patterns. Okay, So one thing we wanted to do is evaluate the performance of this network during periods of running, as well as during periods of stillness. So what we see during running when the rhythm is very strong, as we increase the number of electrodes we're using for our decoding, both our original carrier-based method, the one in which you identify a carrier and use that to demodulate the signal, and this new neural network, which doesn't require a carrier, they perform relatively the same. And this might be expected because the rhythm is very coherent, it's very easy to identify the carrier. And just as a point of comparison, this is what the decoding looks like when you use place cells and you increase the number of cells in your decoder. So, sorry, I should have said this. The x-axis is how many electrodes or neurons you're using, and the y-axis is how accurately you can classify which of the 3 arms of the maze you're on. Chance level being 1 out of 3, 33%. Now, when the animal is not moving, all of these performances drop significantly. Now, as a baseline, we can look at the drop in the performance in the neural decoder. So this might give you some sense of the ceiling, how good we can expect to do based on how the neurons are activating. And we see that the carrier-free approach, which doesn't rely on a carrier, seems to outperform the carrier-based approach. So it's as if the theta rhythm that we see by our naked eye and can identify using PCA, it's not a very reliable reference point for decoding these patterns, because you can do much better when you don't use that reference point. So that's kind of a high-level message that these decoding performances are telling us.
But there one possibility, what is the mic? Oh,
Here.
there one possibility, right? Probably if you have all the neurons working, it's on, and then when you apply the numerical techniques or the mathematical techniques, they are reduced because one cancel out the signal. And then it's no meaning that it's no on. That meaning the signal is canceling one or the ones, but it's on the circuit. It's no off.
So I'll just make sure I understood what you were saying. So you're saying if the rhythm is weak, then the neurons are no longer coherent and their phases are canceling out, essentially. Is that?
Yeah, but the neurons, you can say in this curve, you can say, okay, is most of the neurons is off. No, it's not. The circuit is working probably, or how you know that this is off.
Yeah, so we know that based on the yellow line, which is decoding the activity, the firing rates of neurons. So this yellow decoder is not referencing any timing information. It's just using the spiking of the cells that are recorded with the same electrode array.
But it's after you apply the numerical, the mathematical techniques, right?
No, so these neurons are identified using spike sorting, which happens at a much higher frequency and was done by the experimentalists who we're collaborating with. So they identify the spikes at a much higher frequency. We look at the firing rates of these place cells and we decode the animal's positions using these firing rates. So it doesn't use the pipeline we're using for isolating the theta rhythm.
Thanks.
Yeah, so this drop we really think has to do with the fact that these place cells are just lower fidelity when the animal is still. Okay, so the other kind of cool thing we can do with this network is look at these hidden node patterns of activity during stillness to try to get a better sense of what exactly is going on in the rhythm when these rhythms are weak. So this is what's going on when the rhythms are strong. So I'm showing you 2 different trials, one in which the animal is running on one arm and another in which it's running on a different arm. And I'm showing you 2 nodes of the hidden layer of this network. And what you see is that theta, that is the kind of classical theta, what PCA identifies, is always on, regardless of which arm you're on. So this is a spectrogram, time on this axis and frequency on this axis, and this band means you have a strong 8 Hertz signal in the average local field potential. And what we see is that these hidden nodes, they also seem to have a strong band at slightly higher frequency. So this is happening at roughly 9 Hertz rather than 8 Hertz. You can see this, but by looking at the merge of these two signals, the red is slightly shifted upwards. And this is to be expected because this hidden node is undergoing phase precession, and that implies that it's oscillating at a higher frequency. The colors here show the phase of this hidden node relative to the first principal component. So this phase drift corresponds to the phase precession in the LFP. And if you look at the activity on a different arm, you'll have a different node activate. So this node is learning the information corresponding to this track. This node has learned the information corresponding to this track. And so they're very selective based on the animal's location. Now what we're going to do is look at the activities during stillness. So during stillness, the average theta rhythm becomes much more irregular, as seen by this drift in its center frequency over time. So this is a long stretch of time when the animal is drinking water, and during this, the theta rhythm is kind of drifting as seen in the spectrogram. And what's more, these kind of place theta components that we've isolated using the neural network are also drifting over frequency and over time, with their relative phase relative to the theta rhythm also drifting over time. And this might give a sense as to why you can't use theta as a reference, because its phase relative to these place rhythms is not very reliably maintained over time. It's as if we're picking up two different populations of activity using either PCA or the neural network, respectively. Okay, so I think the last set of results that I'll show you before we can hopefully have more of an open discussion is around trying to connect these different rhythms to different components of the circuit. So I told you before that we have place cells. These are these pyramidal cells in black, which are more localized within the hippocampus. And then we have these interneurons, which are shown in these colored traces that are more distributed within the hippocampus. And if you look at the firing of either these interneurons or these place cells, the interneurons tend to be active in a much more broad sense. They're just always on, regardless of where the animal is in its environment. Whereas the place cells are much more localized in their patterns of activity. And this is kind of reminiscent of what we see in the brain waves, because depending on the technique you use to extract the waves, you either see a signal that is constantly on, or you see a wave, an oscillation that is much more localized in position. And so what we did then is to try to predict the activity of all the neurons that we recorded in our session, and compared the predictions of interneurons to the predictions of the place cells. And the basic idea is that we would expect that the interneurons are more correlated with this general theta that you see going on across the session, and the place cells would be more correlated with these more localized patterns of theta. And that's indeed what we see. If we look at the selectivity of the interneurons, either during running or during stillness, we see that the interneurons are more correlated with this global theta, and the place cells are more correlated with these place thetas. So we take this as evidence that these patterns that are being learned by the neural network correspond to these large-scale assemblies of these neurons that are active at specific positions of the animal's environment. So to summarize, we can use the strongest wave in our recordings as a so-called carrier wave that allows us to decode the signal. But this carrier wave becomes less reliable when the animal stops running. Then a neural network can bypass the need of this carrier wave to identify informative oscillations. And we believe these informative oscillations are being generated by large assemblies of place cells. And at a high level, what all this means is that these patterns of waves, at least in the hippocampus, can convey the precise content of internal states in a variety of these oscillatory regimes. I think in the interest of time, I will skip over the work where we look at noise correlations using this technique. I'm actually happy to stay later for anyone interested, but I will stop and just thank the students that I'm working with. I'm in an undergraduate institute not so far from you all, and I'm happy to take any questions.
Thank you very much for your talk. Well, very interesting.
Thank you.
Thanks.
So something a bunch of us are excited about here is thinking about also not instead of thinking about oscillation, thinking about traveling waves. How does the way you're thinking about oscillations differ? Can you talk about contrapose those two ideas?
Yeah, so there's, I think, a very important distinction that comes up in the context of multi-electrode recordings is looking at oscillations based on their variance versus looking at them based on the information that they carry. So when you see a traveling wave, you'll see a wave kind of sweep over the electrode array. That is, you could say, a very high variance component of what you're seeing because you're seeing the same wave sweep across the electrodes. At the same time, it actually isn't carrying a lot of information, which corresponds to the time-varying qualities of the signal of the animal's experience. And so what we're looking at is kind of actually ignoring these very high variance characteristics, which actually tend to be concentrated in very few dimensions of the signal. So if you have 64 electrodes, you're observing a 64-dimensional signal. The traveling wave is actually inhabiting like one or two of those dimensions, the stuff that you see by your eye. The information that we find in these waves, in contrast, is distributed very strongly across all 64 dimensions. So you can even throw away the traveling wave and you'll still get all of these very nuanced fluctuations that are invisible to the eye, but carry a lot of information. So I would say what we're seeing using these techniques is actually fundamentally different than what people report using when they see traveling waves.
Do you think thinking about a traveling wave as a carrier wave would be a useful way to like recenter things and maybe have a different readout?
Yeah, so when we apply PCA to the multi-electrode signal, we're applying PCA to the complex valued signals. So that means that the first principal component is actually capturing the phase relationships across electrodes of the dominant component. And so even just one component can capture the traveling wave. So the carrier that we use is that traveling wave that you see by eye. But it doesn't actually influence or inform the math that it is a traveling wave. That part is kind of ignored because we're not preserving any anatomical information with respect to the electrodes.
Hi.
Hello.
Can you hear me?
Yeah.
I enjoyed your talk. I was just wondering if you could just say something briefly about your third topic, the representation drift. I'm quite curious what you found there.
Sure. If you're interested, I could spend like 5 minutes doing it. I'm wondering if there's anyone who has like a burning question, or if you have like a time that you need to be out by, then I could just go there. But if there's any final fast questions before I go over the third part. Okay, so then I'll show the third part, and I'll try to run through it pretty fast.
Cool, thank you.
Yeah, thank you. So the basic idea, and this is a senior thesis project of a former student, is that if you look at patterns of activity in the brain, you can use them to decode the stimulus, but one issue is that if you use the same stimulus many times, there's noise in what you see. So the response shifts on a trial-by-trial basis. So we can look at noise in these recordings by looking at these place representations and seeing how they change on a trial-by-trial basis. So what I'm showing you now is I'm adding another axis, which is the trial number. The animals run the same maze about 30 times. 3 times 30 is 90, so we have 90 trials. And what we then do is plot the response as a function of trial number. And what you might be able to see is that these place fields, if they were purely uniform or consistent, you would see these vertical bands corresponding to the place field being fixed across trials. And instead, what you see is that these place fields kind of slowly drift across trials. And so this is kind of interesting when you think about what that implies about the noise in the hippocampus. So this drift that we see across trials can be modeled as some sort of drift in the place fields of individual place cells. And so you could imagine two different general classes of model, one in which the place fields of individual cells drift independently to each other. So that would be if they were kind of they didn't influence each other. And if you have this kind of independent drift among place cells, because the LFPs are averaging over potentially thousands of cells, then on average, the drifts across cells would cancel each other out. And what you'd be left with is a more uniform pattern in the LFP that cancels out the drifts of neighboring cells. In contrast, you can imagine a scenario where all the cells' drifts are kind of coupled to each other. And when you have a system like that, when you have correlated noise, then the drift at the level of the LFP will also show that, because you're averaging together thousands of neurons, all of which are experiencing a consistent form of drift. So the prediction, based on what we see at this level over here, is that individual place cells will all drift together as if the entire map of space within the animal is drifting in this coordinated sort of way. And so we verified that by looking at the drift of individual place cells, and that's indeed what we see. And what's more is that we've found a recent study that actually really corroborates this way of thinking about things. And this is a theoretical study which actually models place activity in the hippocampus. And their basic idea is that if you have an animal that's navigating an environment, then you can think of the patterns of the population activity of the cells of this animal as inhabiting a manifold that corresponds to the environment that they're in. So if you're navigating a 2D environment, then the population activity of your place cells will be constrained to some sort of two-dimensional manifold in neural space. But in their view of it, the thing that this manifold is trying to preserve is not where each cell activates, but rather the pairwise distances. So if two locations in the animal's environment are very close to each other, you would want a neural code that has relatively similar patterns of activity for these two different locations. And so as long as this metric of the outside world is preserved in your population code, then you are doing a good job representing the world. And you can actually implement this objective using a relatively simple form of learning, a simple neural network that learns based on to maximize this objective or minimize this objective. And when you look at the activity of the neurons in this circuit, you actually see that all of the neurons kind of drift their map in space, a phenomenon known as representational drift. And the idea is that it's okay for the code to drift in space because you're still preserving this metric of the outside world. And so you're kind of drifting along a degenerate space of equally good solutions. And so the drift that we're seeing in our maps over here might correspond to the population code drifting in this degenerate space. That's the kind of high-level summary of this.
Hi, thanks. That was pretty. That's pretty interesting. I have two more questions.
Sure.
One is you talked about intertrial variability, and then you have drift. Do you have an idea of the time frame? Are they both the same, or drift is over a longer time frame?
Between which two things?
At the start, you had said there is intertrial variability. I'm assuming that's so that the trials are pretty close in time, but drift seems to be over longer time frame.
Yeah, so this drift is happening on the time scale of minutes, and they're actually modeling this on much longer time scales, like kind of days. So the kind of related to the earlier question, like how many days over which this map is preserved, in their view, the whole map is drifting over days. Now, this is actually kind of the main form of variability that I see. So it's happening at the level of subsequent trials and happening over the timescale of the entire session. I don't know if that addressed your question. Is that getting at it?
Yeah, no, I just wanted to get an idea of like, because I've read about drift in other contexts too, and they usually talk about it over a matter of weeks. So
yeah.
I was trying to relate the intertrial time frame to time frame of weeks.
Yeah, I think it's hard. Part of the reason it's hard for us to look at that, for the same reason it's hard to match cells across days of a recording. The patterns of LFPs that we're seeing are so dependent on these very nuanced high-dimensional patterns that I think across days it's harder to validate these maps.
Yeah, that makes sense. And I was also curious if you've thought about is there any advantage to drift, or what purpose it could be serving?
Yeah, so I mean, the hippocampus, its job is, one job it's kind of touted to play is to write new memories. So one might think that this is just kind of a running average of where you think the map should be based on your recent experience. I think their argument, the theoretical argument that they might make is that it doesn't really matter whether the map is here or shifted a little bit because you're still doing a good job preserving distance. I think one other thing I'm really interested in looking at is how these relate to what are called ripple or replay events in the hippocampus. which happen when the animal is still and represent like massive bursts of population activity that simulate the animals running through the environment. So I'm curious if those nudge the map around every time there's a ripple.
Okay. Thanks. I enjoyed your whole talk, and thanks for going over the representation of drift part also.
Thank you. I appreciate it.
Okay, so I think we'll stop the recording now. And if you want to hang out maybe, or you can disappear or whatever everybody wants to do.
Cool. Yeah, I'll hang out a little bit.